Assalamualaikum and a very good day to all. Today, I would like to present the findings from our research entitled The Changes of Phenolics Profiles, Amino Acids and Volatile Compounds of Fermented Seaweed Extracts Obtained through Microbial Fermentation. I'm Norakma binti Muhammad Noor from University Selangor. Okay, for the introduction, in this study, we are using Cophaficus species, which is a type of red seaweed. Why we choose seaweed? Okay, because it is a good source of bioactive compounds. It has biomass and also biofuels. In Malaysia, Cophaficus species are found abundant, especially in Sabah coastline. Why we choose microbial fermentation as the method to extract the bioactive compounds in seaweed? Because through microbial fermentation, it can improve nutritional value and product characteristics. It is an interesting alternative to conventional method of extraction such as solvent extraction. Okay. Previous research indicated that fermented seaweed extracts enhance in phenolic content and antioxidant activity. Therefore, the composition and profiling of fermented seaweed extracts is very important to be determined before these biomaterials can be used in food or cosmetic industry. Therefore, the objective of this study is to investigate the effects of solid state fermentations on the phenolic species. Okay, for the materials and method, there are 3 samples used in this study, which is GF sample or green flower sample, PG sample or purple giant samples, and WG samples or white giant samples. Okay, the starter culture used is the Aspergillus oryzae, and solid state fermentation was conducted at optimum condition at 30 degrees C for 4 days, 70% initial moisture content with 10% inoculum level. At the end of the fermentation, 100 ml of distilled water was added and agitated at 180 RPM for 1 hour, followed by centrifugation and filtration. Okay. 3 analyses were conducted, which is LC-MS-MS to determine the phenolics profile, HPLC to investigate the amino acid composition, and GCMS to determine the volatile. For the results and discussion, for phenolics profile by LC-MS-MS analysis, the most significant findings is the presence of caffeic acids in all samples investigated, which was enhanced through fermentation. It was noted that caffeic acid in GF samples was increased by 1.7%, while WG enhanced to 123.5% and PG was 20% increase after fermentation. Table 1 shows the phenolics profile detected through LC-MS-MS for all samples before and after fermentation. And this is the full MS scan for all samples before and after fermentation. Okay, this is a summary of the percentage of enhancement or decrement of phenolic compounds after SSF. Okay. That show an increase in total amino acid after fermentation. Most seaweed species contain all the essential amino acids and are a rich source of the acidic amino acids, such as aspartic acid and glutamic acid. According to Ziran and Amino 2017, amino acids in Cophaficus species constituted mainly aspartic acid, glutamic acid, proline, alanine, and leucine, but lower in histidine. Interestingly, in this study, all samples contained lower in histidine, but it was increased after fermentation. And the highest aspartic acid was found in GF samples after SSF. However, aspartic acid was decreased in WG sample after SSF, and PG sample show a slight increment in aspartic acid from 0.02 to 0.52 gram per 100 gram protein after SSF. Or and after solid state fermentation. And for the volatile compounds, it was found that hydrocarbons is the major group content. It plays a vital role in sexual reproduction of the species, for example, heptadecane. Icosane was found significantly reduced after fermentation in WG samples, while GF and PG sample show only slight increment after fermentation. There are also other ester compounds detected, such as pentadecanoic acid and octadecanoic acid. And table 3 shows the volatile compounds detected in GF, WG, and PG before and after fermentation. So in conclusion, phenolics profile, amino acid composition, and volatile compounds of Cafeficus species were changed due to microbial fermentation. Amino acid composition increased to LCMS-MS, and volatile compounds detected to GCMS show some compounds were increased while some other compounds were decreased by SSF. Cafe acid was found to present in all sample used and also enhanced through SSF. Therefore, the findings from this study are very beneficial as it is needed before these biomaterials can be manipulated as food or cosmetics ingredients. So that's all. Thank you.